Assalamu alaikum and welcome back to another video. In today's video, we are going to discuss about a process of DNase footprinting. Now, in this technique, we are going to look at the interaction between the DNA and protein. Now, if you remember in the previous video, we also discussed the technique of electrophoretic mobility shift assay, and that was also to detect the interaction between a protein and a DNA. Similar cases here, but here the technique is a bit different. Now let's go through the technique of DNase footprinting. Now, first and foremost, we start with a double-stranded DNA. So you take the double-stranded DNA and we label one of the strands of the DNA. Now this orange right here dot, this represents the labeled DNA. Next step is we bind our protein of interest to this labeled DNA. Okay, so we allow the proteins to bind, we allow that protein to bind to its target DNA. Now you can see this has formed a complex with the DNA, and then what we do is we digest this DNA protein complex under the mild conditions with the DNase 1. Now, what is the function of DNase 1? Obviously, what it will do is it will chop this DNA on the right and on the left, sparing the region which is bound by the protein. So, in this region where you can see the yellow, which is a complex of DNA and protein, the DNase, it will not be able to bind or it will not be able to cut the DNA because now the protein, it's basically protecting the DNA. But in this region on the right side and on the left end, it will break or it will cleave the DNA. That's what we are seeing in the next step. So we will follow it by removing the DNA and then denaturing the DNA. Now what we are going to see is the fragments. Now obviously all of the fragments here we have are end labeled because we have already performed the labeling of one of the strands. Although we will get fragments after denaturing, we will get fragments from both of these DNA strands. But since we have labeled just one of those, all of the fragments which we will see, it will be of the same. Okay. So what you can simply see from the diagram here is that this DNase is able to digest at different fragments except the region here, and that's what we are seeing here in this region. Okay. So now till now it has yielded us those end labeled fragments. Now what we'll do is next we will proceed with the electrophoresis of these fragments. So those labeled fragments, these are then set for the electrophoresis, and we perform the autoradiography in order to detect those fragments. Now, another thing is when we are performing electrophoresis, what we can do is we can also perform it under the conditions where we have added no protein. So the lane 0, it shows us where you have added no protein to it. So obviously, if there is no protein, there will be no protection of that DNA. So obviously the DNase, it will now be able to easily cleave the DNA without protecting any significant portion of DNA. But in the second case where we have added, now what we are doing is we are increasing the protein concentration. So in here you have added 1 unit of the protein. So what we see is slight protection. Although we are getting bands in that region, but we see this region, it is protected by the complete action of DNase. And that protection is because this protein is binding in this particular region of the DNA. Similarly, when we proceed ahead and we add 5 units of protein, now if you are going on adding higher and higher amount of the protein, what will happen is you will see a region here which is completely protecting the DNA from the attack by the DNase, and this region we call as the footprint. Now, when you get to this point where you are equally able to see that this region is protected, now you can easily detect from that point onwards that now this is the region where my protein is interacting with its target of DNA. So this technique, it tells us about the interaction between the DNA and protein. So these were two techniques which we have discussed so far. We talked about the EMSA, that was again a DNA protein interaction, but it was on the basis of the so when a protein is interacting with the DNA, again, what happens, there is a change in the shift on, obviously when we run it on a gel. But in this case here, it's the protection of the protein binding to that segment of DNA and protecting it by the action of the DNase. So that's how we can detect the interaction between the DNA and protein.